Alright, okay, so hello dears. Ayan. So welcome back to the second video, yes, of our pre-recorded lecture in our class in Immunosero, and with the topic again, which is your Syphilis Serology. And for this video, we're going now to start with the different methods on how we diagnose, okay, different laboratory methods on how we diagnose syphilis. But before we start, I have to change what I've said. I have to retract my statement. So, it was not syphilis pala ang nadala ni Christopher Columbus from Europe to America, okay? So, it was syphilis pala that Christopher Columbus and his crew got from America, and then may iyang i-transmit to Europe, okay? According mo daw na i-think or i-postulate, okay? So, syphilis ang nakuha sa ni Christopher Columbus and his crew from the Americas, okay, American islands, and then na-transmit to Europe, okay? So, from New World to Old World, okay? Sige. New World to Old World kay syphilis, okay? But from Old World, meaning from Europe to New World, so ang nadala daw ni Christopher Columbus, to the Americas is smallpox. Ayan, sorry. I felt kanina dyan na sa ito. But yeah, so smallpox ang nadala ni Christopher Columbus from Europe to the Americas, okay? But from Americas to Europe, ang i-transmit ni Christopher Columbus to Europe daw kay syphilis, okay? Alright. And aside from that, another one, before I forget, is lumalabas sa boards. Again, di ba, the first treatment to syphilis was your arsenic, di ba? And this drug was known to be, is salvarsan. Hindi siya yung first name. Salvarsan. Okay, and or also known as arfenamide. Okay? And this was discovered by, guess who? Kinsa to nag-coin sa term sa complement? Yes, press the buzzer, si Paul Ehrlich. Siya ang nakadiscover sa salvarsan, arfenamide, the first treatment for syphilis. It was originally, before the name salvarsan, si Paul Ehrlich, iyang gi-test ni, and iyang gi-name as compound 606. Ayan. Nako, please take note, okay, lumalabas sa board. Salvarsan, the first drug against syphilis, or also known as arfenamide. So it was made out of arsenic, okay? And it was discovered by, no other than Paul Ehrlich. And before the name salvarsan, it was known as compound 606. Okay, so it was named compound 606. Alright, so mga additional lang na forget na ako ganina sa first video. Sorry naman. Okay, alright. So, mga katulang to siya. Additional infos, mga must know kasi lumalabas din sa boards. Okay, alright. So, we now go to the different laboratory diagnosis. Okay? So, we'll start first with your direct detection. And for direct detection, diba, is of course, your dark field microscopy, diba? Press the buzzer or recall in your backte, intro to micro, diba? Basta ganyan mga dark field microscopy, when you talk about dark field microscopy, the first organism here, or the first thing that should come to your mind is that it's usually used for your spirochetes. Okay? So here it is now. Makita natin here, dark field microscopy again is used for your spirochetes, direct detection. Okay? So we visualize motile treponins from lesions. Okay? So here is an example, dark field microscopy, diba? So they appear light against a dark background. By the name itself, dark field, ang field kay dark, but sila, they appear light, okay? So they can be seen against a dark background, okay? Alright, and they are the treponins, yes, they are motile from your active lesions, okay? And aside from that, direct detection pa din, you can use fluorescence microscopy. So what you do is either direct or indirect fluorescence immunoassays, diba? They call in our labeled immunoassays. What you do is you then add mga fluorescent probes, or you stain your treponins with fluorescent stains or fluorochromes like TRITC, FITC, diba? So, and then after a while, you then examine under the fluorescence microscope, and they can be seen again fluorescing the color depending on the stain that you use. So it could be again TRITC, FITC, batchmate now, TRITC, FITC, diba? So again, it depends on the color. So this one is apple green, so this is FITC, diba? Fluorescein isothiocyanate. So these are direct detection, meaning they are the sample came from the lesions. So These methods are usually advisable for primary syphilis, okay? Because in primary syphilis, again, the lesions are there, your chancre. So pag swab mo sa chancre or pag get mo sample sa chancre and then you examine under the microscope, there are motile treponines that you can see there, okay? So either, again, dark field microscopy and fluorescence microscopy. For primary syphilis, the serological or your antibodies, they usually develop after mga 1 week, okay? And then 3 weeks na yun siya ma-detect, okay? So for serological tests, it's not advisable that you test serologically here in your primary syphilis, okay? Because the best test, Jude, for primary syphilis, if the syphilis of the patient is still in its primary stages, is through direct detection. So microscopy, dark field microscopy or fluorescence microscopy, depending on the lab that you are having the test. Okay, all right. So that's for dark field and fluorescence microscopy. Now, in the case that you cannot detect any more motile treponines in the lesions, nag-try ka sa lesions, wala yung motile treponines, or there are no any more lesions, but the symptoms of the patient are really consistent, consistent with syphilis, then you go now to the different tests serologically of your syphilis, which are now divided into two. It could be non-treponemal, okay, or treponemal. Alright, so non-treponemal tests or treponemal tests. That's serologically. Okay, so we'll start first with your non-treponemal test. Again, I have to emphasize, it's important, again, and it's mandatory, required talaga, that the patient here, if you are going to examine using microscopy, direct detection, it's important that the patient has active lesions, okay? Because again, it means that the treponines are still there. It's still fresh. They are motile. You can see it in the lesions, or you can see it from the lesions, okay? Alright. So again, if you cannot detect that anymore, there are no lesions, again, you can now use the other tests, serological tests, either non-treponemal or treponemal tests. And we'll start first with the non-treponemal tests. Okay. Now for the non-treponemal tests, what we are detecting is what we call your reagin. Okay, ako na siyang batchmate. Charot. Say reagin, or also known as your non-treponemal antibody. Now this is an antibody to cardiolipin. So cardiolipin siya, it's a lipid that is released from damaged cells. Okay, lipid material from damaged cells, and it's usually seen in syphilis. But aside from that, reagin, again, is an antibody. Reagin is antibody to cardiolipin. So what we're detecting in non-treponemal tests is an antibody known as reagin. Okay, and unsay antigen ni reagin, or what it detects, or what is it against, is your cardiolipin. Now the cardiolipin is, again, is lipid material from damaged cells seen in a lot of infections, not only syphilis. That is why for non-treponemal tests, your non-treponemal tests are not specific. They are not specific, but they are sensitive. Okay? So, we're detecting an antibody to which is the cardiolipin, which is reagin. It's usually seen in syphilis, but not only in syphilis. Okay? Alright, ayan. So, that's why, basta mga non-specific, usually, mga non-treponemal tests, they are used to screen for syphilis. So it's only for screening tests. Okay. Alright, now we have three tests. You have the Vdrl, Venereal Disease Research Laboratories test. You have Rapid Plasma Reagin, RPR. And lastly, the toluidine red unheated serum test, or TRUST. Big word. Okay, alright. Ayan. Now, venereal, you maybe think, what is the meaning of venereal? Venereal means sexually transmitted, if I'm not mistaken. If I can remember it correctly, I think that's the meaning of venereal. It means it can be transmitted sexually, or it's of sexual origin, something like that. Okay, alright, ayan. Now, for TRUST, for TRUST lang, because we're not going to discuss that much. TRUST is a modified test of Vdrl. Okay, so it's a modified test for Vdrl, and it's similar also to RPR, but in this test, TRUST uses red particles. Ayan, by the name itself, toluidine red. So it uses red particles for better visualization of the results. Usually, I think it's still the same principle with Vdrl and RPR, flocculation later. When we go to Vdrl and RPR, they are the same principle, but for TRUST lang, toluidine red, nasa pangalan na, it uses red particles, okay, so that visualization is much enhanced and you can see it better. Okay, that's for TRUST. Alright, ayan, so again, as mentioned, non-treponemal tests use to screen syphilis, but it's not specific for syphilis, but it's highly sensitive. That's why for screening tests, it's best that you want the tests to be sensitive, because screening, you just want to know if the patient really has this disease. Okay, so for confirmatory tests, you want these tests to be much more specific, because you want to confirm na. You want to know if the disease is really caused by this organism. So you want to confirm. So for confirmatory, it should be specific, meaning the test can only detect what you want or can only detect the organism that you want. Okay? Alright, so mga basic concepts. Okay, alright, so non-treponemal tests. So we'll start first with your VDRL, your Vdrl Disease Research Laboratories test. Your specimen is heat and activated serum. So still the same, di ba? 56 degrees Celsius at 30 minutes. Still the same purpose to inactivate complements or your CSF. For principle, you have flocculation. So it's a precipitation reaction with very fine particles. So you cannot see it that much. So that's why later we need to use the microscope, okay, to understand or to look into the results or the reaction. Okay. Now for the reagent antigen is the Vdrl Antigen, and the Vdrl Antigen is composed of your 0.03% cardiolipin, which is the main reacting component. You have 0.9% cholesterol, okay, and 0.21% lecithin. 0.21% lecithin is yung purpose there is to inactivate the complement-like activity of your cardiolipin, if I'm not mistaken. Yes, I will check on it again. I think okay, but yeah, that's the purpose of lecithin. And cholesterol is for what's the purpose of cholesterol? I think it's for attachment of the antigen, if I'm not mistaken, again. Alright, so I have to confirm that. Alright, but these are the components of your antigen, Vdrl antigen. And the main component is, of course, cardiolipin. Okay, alright. Now, for antigen delivery, what we call a Hamilton syringe, Alexander Hamilton, charot lang. There are... Check lang. Okay. Yes, I'm a Hamilton fan. Yes, sorry naman. Okay, anyway. Wait, nag-fangirl ko like mga 5 seconds lang. Okay. Hamilton syringe. Okay. Hamilton syringe in any appearance. Now, your Hamilton syringe doesn't have a needle. Alright? But during, tama? Yeah, it doesn't have needle. But during the dispensing of your sample, it needs a needle. So, you need to inject or put a needle. Yes. With or without the bevel. Yes. So, this is your Hamilton syringe. In itself, it doesn't have a needle, but for delivery of your antigen, Vdrl antigen, and your reagents, it needs to have a needle. So you have to put a needle there. Okay, with or without a bevel. Okay, alright, ayan. Okay, now for rotation, it's 180 RPM, 4 minutes for serum, and 8 minutes for CSF. Now, this is important. Lumalabas sa boards, like how many rotation, how much on rotation. So, for VDRL, it's 180 RPM, 4 minutes for serum, and 8 minutes for CSF. How did I remember that? I think 8 minutes CSF. So I think 8 minutes mas matagal because CSF is in the brain, so mas mataas pa pumunta sa brain, something like that. I think that's how I remember. Or wala ko yung mnemonic for it. Nag-ano lang ko. 8C, mga ganun naman for serum. But anyway, kamo rin bahala sa iyo ang mnemonics. Basta 4 minutes for serum, 8 minutes for CSF. So, it's either you need a mechanical rotator, okay, alright, and you can use it manually, pero mas best yun a mechanical rotator. Okay, alright, okay. Yeah. So for Serum Vdrl, this is your slides, okay, it's ceramic rings, 14mm in diameter. It may be qualitative or quantitative. So for qualitative, if it has a result, you then proceed to quantitative Vdrl. So same procedure natin with our CSF, CRP, diba, na mga latex. If there's a positive, we then dilute, mga serial dilution, and then the highest dilution that contains a positive result is the titer. Okay, we reciprocal, diba? Okay. Alright, so maybe qualitative or quantitative. So it's ceramic rings for Serum Vdrl. And for CSF Vdrl, it's slide with rings. It's much more wider. It's wider iyang diameter, 16mm. And the depth is 1.75 millimeters. Okay, alright. So very important, very particular board exam or mga exams din on the mga materials na ginagamit for serum, CSF, Vdrl. So it's important yun that you know. Okay? Alright, so for serum Vdrl, ceramic rings, 40mm in diameter. For CSF Vdrl, it's not necessarily ceramic, 16mm in diameter. Excuse me, sorry, allergies. Okay, alright, ayan. So serum Vdrl, excuse me again. Sorry, allergic rhinitis. Sorry guys. Okay, anyway, serum Vdrl and CSF Vdrl. Okay, alright. Now, again, as mentioned, because it's flocculation, so very fine particles, so we need to examine it under the microscope. LPO, 100x total magnification. So for the reporting, non-reactive, no clumps. Weakly reactive, small clumps. And reactive is medium to large clumps. Okay, that's for VDRL. Okay, now the next non-treponemal test that we are going to discuss is your RPR, or Rapid Plasma Reagin. Now, RPR is considered to be the most widely used non-treponemal test. Okay, this is the most widely used non-treponemal test, press the buzzer, RPR. But some of the reference laboratories, research laboratories, they tend to prefer the VDRL. Okay, but again, most widely used non-treponemal test, routine or hospitals or whatever, it's your RPR, Rapid Plasma Reagin. Now, for the specimen is your serum. It doesn't need to be heat inactivated. Okay. For the principle, it's now charcoal agglutination. So we use now charcoal. Okay. So the reagents contain charcoal. Alright. Now, reagent antigen is the same with Vdrl antigen, but it's modified. So it's Vdrl antigen, all of those, okay, plus charcoal, EDTA, thimerosal, and choline chloride. And the purpose of the choline chloride is chemical inactivation. Charcoal is used there for easier visualization, kay dagko ang particles, alright? So, therefore, agglutination will be much visible because dagko ang particles na gigamit. EDTA, it prevents the oxidation of your lipids, di ba? Vdrl antigen contains mga cholesterol, your cardiolipin. So, to prevent the oxidation of the lipids, EDTA is there. And thimerosal, ang purpose niya is a preservative. Okay? So, again, ang charcoal for easier visualization, they are large particles, so you can really look or you can really see the agglutination reaction happening. Your thimerosal is a preservative, okay, and your EDTA prevents the oxidation of your lipids found in the Vdrl antigen, mga cholesterol, your cardiolipin, okay, and choline chloride, as mentioned, it's for chemical inactivation. So to inactivate complements again found in the serum and all that. That's why there's no need for heat inactivation because you have choline chloride. Okay, alright. Now for the antigen delivery, it's still the Hamilton syringe. Parehas sila. Hamilton, yes. I have soldiers in the rotation, therefore, now is much lesser ang RPM. 100 RPM na for 8 minutes. That's for RPR. Okay. Now for RPR, it uses plastic cards. So the same may be qualitative or quantitative, and they are now examined macroscopically, so you can see it really. Because again, it uses charcoal. That's the purpose of charcoal. Kaya dagko ang particles, so mas dagko ang reactions may tabo, so agglutination will be much visible. Okay, alright. So here are your plastic cards. Actually, this is the laboratory test that we're going to perform in your syphilis in the laboratory. Okay, and also the kits for syphilis. Yeah. Okay. Alright. Okay. Now for reporting, non-reactive, no clumps, weakly reactive, small, black clumps, and reactive, medium to large, black clumps. Again, you are detecting reagin, both VDRL and RPR. Okay, so, but again, the downside lang is it's not specific for syphilis because reagin can also be found in a lot of other diseases, like lot of diseases. Okay, so pwede siya sa rickettsial diseases, pwede siya mga hematological diseases then. So it's very, very non-specific. Okay, alright, ayan, so RPR. Okay, now, if you have a positive treponemal test, a non-treponemal test, we then subject your sample to a treponemal test because the treponemal tests now are your confirmatory tests. Okay, but they are more difficult to perform and more time-consuming. Now, for the treponemal test, we detect now antibodies against your Treponema pallidum, and again, usually used for confirmation than for screening because it's much more specific. You are detecting now antibodies against Treponema pallidum, not reagin, which is a lot, diba? It can be detected in a lot of disease and reagin. But for treponemal test, you detect anti-TP. So antibodies to Treponema pallidum, TP. Okay, and we have different tests. You have Treponema pallidum immobilization test, TPI. You have the fluorescent treponemal antibody absorption test, FTA-ABS. And you have the heme agglutination or particle agglutination tests. Again, usually used for confirmatory tests. Okay, so we start first with your Treponema pallidum immobilization test. This is considered to be the test to which the other tests are evaluated. Yeah, so it could be said that it's the reference method, siguro, if I'm not mistaken, ha? Okay, but it's the test to which the other treponemal tests are compared to, okay, or evaluated to. Okay, now what is the procedure? You have a live T pallidum. It came from a testicular chancre of a rabbit, diba? So, inoculatan ang rabbit of T pallidum, and then it created a chancre, and then you get the sample. Live T pallidum there, plus the patient serum, which we now expect to have the antibodies to Treponema pallidum. And of course, what happens is the antibodies in the patient serum will now attach the live T pallidum, leading to immobilization. Hindi na siya mulihok, okay? So the positive result under the microscope, dark field microscope, if greater than equal to 50% of the organisms are immobilized. Okay. So for interpretation, positive, greater than or equal to 50% organisms are immobilized. Doubtful, 20 to 50%. And negative is less than 20%. So when you say doubtful, it could be that gamay pang antibodies, alright? Or it could be that it's not really syphilis yun, maybe it's just a biological false positive. Okay, it could be caused by other diseases, diba? Or whatever. Okay, so it could be that you need to retest the sample after a week para more antibodies are formed, okay? Para to really verify. Okay, so that's the TPI, Troponin Palindrome Immobilization Test. But this uses dark field microscope. So it's time consuming and it's quite expensive then. Alright, so that's Troponin Palindrome Immobilization Test, but the name itself, immobilized. So this lang mulihok because of the antibodies found na in your patient serum. Okay, again, it's the test to which the other treponemal tests are evaluated to. Okay, alright. Okay, so for the next treponemal test that we're going to discuss is your fluorescent treponemal antibody absorption test. Okay, but before we continue, ayan, so I now remember cholesterol, the purpose of cholesterol in your Vdrl antigen is that it improves or enhances the reacting surface of your cardiolipin. So sa surface of cardiolipin, it enhances the reacting capability of it to your reagent or to your antibody. Okay? And salvarsan also, before I forget, is also coined as your magic bullet. Ayan. Diba? Magic bullet. Because I think pag discover na sa salvarsan, pack na ayun ang syphilis. So magic bullet, ang other name niya. And also, what is considered to be the first serological test that was invented to detect syphilis? Nako, preso buzzer, it is your Wassermann test. Wassermann, tama. Okay, Wassermann. The Wassermann test, which is attributed to Paul von Wassermann. Ayan. So, siya ang wrong spelling. Wassermann. Siya ang naka-discover. And this principle is based on, familiar ang Wassermann test? Asa man na-handout nato siya nabutang, it's in your complement fixation. So, it follows the principle of complement fixation test. Ayan. So, aware na, di ba? I hope na-gets lang ang principle sa complement fixation. Okay? Nagawa sa inyong quiz. Yes, pina-essay. Explain the principle. Okay? Alright. So, di ba, for complement fixation, so Wassermann test, we're detecting antibodies to syphilis. So, therefore, reagent antigen contains syphilis antigen, so you're detecting antibodies to syphilis. So, pag-add nila, di ba, if the patient already has antibodies to your antigen, okay, so meaning na siya syphilis antibodies, so the reagent syphilis antigen, so mag-react sila, okay, they form an antigen-antibody complex. Pag-add sa imong guinea-pig complement, so ma-fix ang complement there, di ba, because complement is activated if there is antigen-antibody complex formation, di ba? So, ma-fix ng complement there. So, pag-add sa sheep RBCs with hemolysin, okay, so, dili na siya, wala na yung complement na makabind sa imong sheep RBCs naman na hemolysin. So, therefore, the sheep RBCs will not hemolyze. So, that's the positive result, no hemolysis. So, what does that indicate? A positive result, no hemolysis, indicates that the patient has the antibody to the known antigen. So in this case, the patient has syphilis antibodies brought about by infection with syphilis. Okay, so I hope na-gets na kayo, diba? Essay to siya sa quiz and medyo may mga na-discover ako na parang, weird. Basta. Okay, tapos, sige lang. I will try my best, or parang I just let it slide na lang. I tried not to be stressed about it. But sige lang. Okay, alright. So, I hope na-gets lang. Okay? Alright, so that's the principle of complement fixation. Wassermann test, that's the principle. The first serological test to detect antibodies to syphilis. Okay? Press the buzzer. Ayan. Alright. Now, we go na to your fluorescent treponemal antibody absorption test or your FTA-ABS. Okay, CBN, charot. Okay, so it's one of the most used confirmatory tests, an indirect fluorescent antibody test. Yes. Basically, it's really used for confirmatory, very popular for confirmatory tests. Very popular as a confirmatory test ay yung FTA-ABS. Okay, now what is the principle? We first inactivate the patient's serum, again, to inactivate your complements because it may cross-react, no? It may interfere with the test. We then add a sorbent, which is your writer's strain of T. pallidum. The writer's strain, yes, is a non-pathologic strain of your T. pallidum. So why do we need to sorbent? It's because we want to remove cross-reactivity, okay, of your syphilis antibodies or any your antibodies to syphilis to other Treponema pallidum subspecies. Okay, so we just want to make sure yun that masorb siya daan diri sa imuhang writer strain of T. pallidum. Okay, because again, we don't want any cross-reactivity. So, meaning, imo sang ipareact daan ang imuhang antibodies, okay, sa imuhang patient serum with a strain of T. pallidum para gyud mawala itong mga other cross-reactivity, mawala itong other interferences. Okay? Alright. Ayan, the sorbent removes cross-reactivity with treponines other than T. pallidum. Nara din ang explanation diri, yes. Okay, alright. So, we first absorb, no? Or we use a sorbent, which is a writer strain. And then after, the nickel strain of T. pallidum, this is now your pathogenic strain or pathologic strain of T. pallidum. We then add with your patient serum, which is now already absorbed, no, or with already underwent sorbent, the sorbent procedure with your writer strain, and we add also a FTC labeled AHG reagent. So this is a fluorescent, yes, fluorescent test that uses AHG, diba, with a FTC label. Okay, and then we then look for fluorescence. Ayan, so that's a procedure. By the name itself, fluorescent treponemal antibody absorption test. Okay, so we're looking for the antibody to Treponema pallidum using fluorescence. Okay, using fluorescence, FTC labeled AHG. Okay, all right. So for grading, 0 non-reactive, 1 plus minimally reactive. You repeat after 1 to 2 weeks, and then 2 plus and 3 plus to 4 plus is reactive. So you look at the intensity of light. Okay, so I think naran siya. I think it would be by the machine or the computer siguro attached to your fluorescence microscope. All right, and or it has already a parang I think na siya mga each of the positive results or kani mga results na siya own parang grading niya or like kanang counterpart, yes, na siguro wavelength ba or intensity of light. Okay, and that's how you grade. Okay, basta 2 plus to 4 plus is considered to be reactive. 1 plus is minimally reactive pa lang. You repeat after 1 to 2 weeks. It could be that wala pa kay antibodies na na-produce. Okay, so you repeat after 1 to 2 weeks. And then 0 is non-reactive. Okay, alright. Now before we proceed, guys, please take note that for the non-treponemal tests, RPR, again, is the most widely used serological test and is also considered to be more sensitive than Vdrl in primary syphilis. Okay, so it could be that, diba, as mentioned, primary syphilis, wala pa kayo serological tests or wala pa kayo serological response ang body to primary syphilis. But diba, after a week, pwede na siya mag-develop of antibodies. So if you want to detect that, no, more sensitive is your RPR, okay, than your Vdrl. Okay, please take note. That's according to Turgeon. Okay, alright. Ayan, so that's for FTA-ABS, Fluorescent Treponemal Antibody Absorption Test. Okay. So this test again is considered to be the test to which the other treponemal tests are evaluated to is your TPI, Treponema Pallidum Immobilization Test. Okay. Alright, now we go now to the last type of your treponemal tests, which are your heme agglutination or particle agglutination tests. So by the name itself, they are heme agglutination. So you use RBCs. So it's passive heme agglutination test. Ayan, passive heme agglutination test performed in microtiter plates using RBC sensitized with nickel strain. So that's why it's passive agglutination. It's because the RBCs do not contain the nickel strain antigens naturally. So it's not naturally found in the RBC antigens. So gi-absorb siya on RBCs. On RBCs, that's why it's called passive agglutination, diba? Okay. Ayan, so I'm sure, I hope lang yun na na-get ninyo ang differences atong agglutination reaction. Sana naman. Okay, alright. So we have different types. You have heme agglutination treponemal test for syphilis or your HATs, which uses 30 RBCs. You have your microheme agglutination assay for antibodies to T. pallidum MHA-TP, which uses sheep RBCs. And your T. pallidum heme agglutination assay, which is your TPHA. Okay, alright. Ayan. So, how do I remember? HATs, maraming T, di ba? So, char. Marami siyang T. Okay, so therefore, it uses T, turkey. Ayan, so HATs, turkey, and your MHA-TP na ang sheep RBCs. Ayan, that's how I remember lang. HATs, daming T, dalawang T, HATTS, dalawang T. So it uses T, turkey, alright. And your MHA-TP uses shape RBCs. Alright. Okay. Now, we have much more recent na test, which was your T-Palindrome particle agglutination test. For this test, it uses gelatin particles instead of RBCs. Okay. Now, these gelatin particles, they are coated with treponemal antigens and they are more sensitive in detecting primary syphilis. That's according to Steven. So again, as mentioned, primary syphilis, the best is direct detection. Okay, but in the case that it's still primary syphilis but there are no motil troponins na, or it's already possible serological response ang body, then you can use na mga serological tests. Okay, alright, and TPPA considered to be more sensitive in detecting primary syphilis compared to the other methods. The patient serum or plasma diluted in, you use microtiter plates and then you incubate with either this T-Palindrome sensitized gel particles or unsensitized particles as a control. So you need a control. Okay? Alright. And then, the process of T-Palindrome antibodies, of course, if there is antibodies to T-Palindrome, there's agglutination that will happen. And this agglutination is manifested as a smooth mat. Okay? Yeah, covering the surface of the well. So there is a smooth mat. Okay, covering the well. And if there's the absence, negative, the gel particles will settle to the bottom of the microtiter plates and they will look like a compact bottom. Okay, so for picture purposes, ayan. So as you can see, this is a mat, meaning smooth mat na covered yun niya tanang surface, tanang area of your microtiter well. Okay, and this is the negative result. So therefore, row C, this is the patient serum. Ang A is the positive control. Letter B is the negative control, and letter C is the patient serum. So kaning column 3 ang mga testing parameters, and then ang 4 kay ang control. Okay? So the row A is positive control and a negative control, and then B is positive control and a negative control, and then letter C, row C is the patient serum. So as you can see, patient serum column 3, ayan, is smooth mat, so suspension gid siya na na-cover ang entire area or sa ilalong gid sa imuhang microtiter plate. So therefore, it's considered to be positive. Okay, alright. So that's for TPPA. Alright. Okay. Okay, alright. Now for heme agglutination tests, again the principle, sorbent-treated patient serum plus RBC sensitized with nickel strain, it leads to heme agglutination, which is now your reporting, which leads now to reporting which is positive. And the negative is no agglutination. So very easy lang. Okay, alright. Ayan, now for the next video, we'll now go to the remaining technologies in syphilis testing. Alright, and until the end, with the different algorithms, testing algorithms for detecting syphilis. Okay, so I'll see you on the next video. Alright.